Hello Anatomy, happy Saturday morning. I just finished Talon, actually it's Saturday afternoon, and as promised I am going to record a lecture for you on skeletal muscle, the physiology of skeletal muscle contraction, and I hope this is helpful to you. If you have questions after I'm done, then send me a remind and I will answer those questions for you. Here we go. I'm going to start over and recap everything that we had talked about in class. So we start off with the gross anatomy of the muscle, the overall structure of the muscle. And so when you look at a muscle as a whole, you're looking at the body of the muscle, and that's wrapped with a membrane. The overall membrane around the muscle is called the epimysium. Okay, and then if you pull out a section of muscle, that's called a fascicle. The fascicle is a bundle of muscle fibers. So I'll write bundle of muscle fibers. And the wrap, so I'll write wrapped with. So the muscle is wrapped with the epimysium. The fascicle is wrapped with, and let's see if you remember, wrapped with a perimysium. The bundle of muscle fibers is really a combination of a bunch of myofibrils. I'm back. I had to pause for a minute because they came in and emptied our trash. So the perimysium wraps a fascicle, and a fascicle is a bundle of muscle cells. Each muscle cell has its own membrane around it, but because it's a muscle cell, we don't call it the cell membrane or the plasma lemma. We call it a sarcolemma. Okay, around the muscle fiber. And in between all the muscle fibers, okay, so between the muscle fibers, we have the endomysium. So the sarcolemma is the wrapping around an individual muscle cell, and here we see it here. And then the space around the muscle cells is in the endomysium. So these are going to be words that you're going to see on your test, okay? The myofibrils are going to be the units of actin and myosin. So they're units within the muscle. And then we have the myosin and the actin, thick and the thin filaments that actually are going to slide past each other to cause the overall shortening of the muscle. So here's another picture of it showing us the actin and the myosin. Again, I said that you needed to pay attention to the position of the Z lines or the Z disc, they're in blue, and then the arrangement of the actin and myosin. So zooming in closer and closer, what's important on this chart? We have these openings for the transverse tubules. Remember, transverse means they go little tubes that cut in across, perpendicular to the sarcolemma, which allows for the calcium to be released, or the stimulation of the action potential, the stimulation of the sarcolemma going all the way down into the interior of the muscle cell, getting close to all of the contractile units, and then releasing the calcium from the sarcoplasmic reticulum, which is just the fancy muscle endoplasmic reticulum. So the functional units are called sarcomeres. The myofibrils are full of those sarcomeres. And the process for contraction of a cell muscle, it's called the sliding filament model, in which these thick and thin filaments slide past each other. The myosin actually pulls on the actin and slides the actin over the myosin. And what you need for this to occur is lots of calcium and lots of ATP. And we're going to be talking about how this process occurs. Here's your simple drawing of a sarcomere within a myofibril. And when you work out and your overall muscle strength increases, that's because you have an increase in the number of these contractile units. Sorry. I woke up at about 5 this morning, so I'm already tired. Here's an up-close view of your myosin And remember, those myosin is thick. Myosin is thick, and those actin filaments are thin. And you can see in this diagram how the Z lines, or the Z disc, they actually get closer to each other when a muscle is contracted. And that's because the degree of overlap between the actin and the myosin increases. An even closer view within one sarcomere shows the thick filaments with these little heads that look like golf clubs. So here we have a thin filament of actin, and you'll notice that there is this protein called tropomyosin, and the protein is laying over these little darkened spots here, and those darkened spots are the myosin binding sites. This is where the myosin would attach, but it's being covered up by the tropomyosin. Then you have this troponin over here, another protein. When calcium bonds to that's supposed to be a 2, calcium bonds to the troponin, it's going to pull and cause the tropomyosin to move away, exposing the actin binding sites. And when the actin binding sites are exposed, one of the heads is going to bind to the myosin head is going to bind to the actin, and the other one, that's the ATP binding site. So there are 2 heads on the myosin filament. So again, reviewing this is our neuromuscular junction, the point where the neuron and the muscle meet at the end of our motor neuron. And notice they don't actually touch. There's that space there, but into that space, when the nerve is stimulated, it's going to release its neurotransmitter. In this case, the neurotransmitter is ACH, which stands for acetylcholine. So acetylcholine, when it's released, it causes sodium and calcium to move across the membrane. You see here, calcium is taken into the nerve, okay? And that wave of electrical potential travels down across from the neuromuscular junction, okay, from the motor unit down the T-tubule. When the when the membrane, the sarcolemma, is stimulated, when that action potential travels down, and this is a chemical electrical charge, calcium spills out of the sarcoplasmic reticulum, which is just endoplasmic reticulum found inside of a muscle cell. When the calcium ions spill out, they bind to the troponin. And remember, the troponin is going to change shape, and the tropomyosin is going to move and expose the active binding sites on the actin. When the actin binding sites are exposed, the myosin cross bridges form. And then they complete what's called a power stroke. In order to stop this process of the contraction, calcium has to be removed from the sarcoplasm, okay, the cytoplasm inside of a muscle cell, so that when the calcium is gone, ATP can bond and the myosin head is going to let go. Otherwise, your muscle is going to stay in a constant state of contraction. So here again, the myosin head attaches to the actin. So this is the actin, this is the myosin. And when the myosin binding sites are exposed on the actin, the myosin head bonds to the actin and does what's called a working stroke or a power stroke and pulls the actin fibers across over the myosin. Then when ATP bonds, this allows for the breakdown of ATP, allows for the myosin head to let go. This is important because you have to have ATP to allow for the muscle to stop contracting. And this is what leads us into what happens upon death. So here's another diagram, another picture view of what happens. And then we have it here in words for muscle fiber contraction, and then muscle fiber relaxation. Here's a new term, acetylcholine esterase. This is an enzyme that's going to break down acetylcholine, which stops the wave of contraction, the action potential across the membrane. So then the calcium ions are retaken back into the sarcoplasmic reticulum. ATP comes in and causes the actin-myosin linkages to break. The cross bridges detach and they recock. That means they get back into their position to be ready to contract again. The connections between the myosin and the actin are broken. And the muscle fiber relaxes, which means that the Z lines that were closer together now slide back into their position where they're farther apart. Here's another picture. I want you to have lots of pictures where you can see this in different ways. So a relaxed muscle versus a contracted muscle. And this is another image of what happens when the triple myosin moves away from the myosin binding sites on the actin. Don't forget, there is that article or that digital textbook available to you on Google Classroom that you can look at as well. And some links to YouTube videos. So rigor mortis, post-mortem, this means after you die. When you die, if your body, or when a person dies, if the body is in a certain position, notice these two bodies, actually the legs are stiff, and here in the bottom picture, the arms are outstretched. When a person dies, because they don't have any more ATP being generated in the cells, those cross bridges cannot release. The myosin heads cannot release from the actin. And so the body, after a period of time, it's going to start with the smaller muscles in the face first. The body's going to stiffen and in this position called rigor mortis, death rigidity. And this is one of the ways that you can determine how long a person has been expired. So this is why sometimes people die and their eyes are open and you can't get their eyes to close. After a while, after a long enough period of time, the muscles do start breaking down chemically. The actin and the myosin proteins begin to decompose and break down. And at that point, the body goes flaccid or limp again. So this situation isn't a permanent situation, but when someone has been dead for 12 to 24 hours, the body is going to be stiff. And there you see two pictures. So that's just a real quick introduction and overview. I really strongly urge you to read about creatine phosphate. There is an essay in your textbook about it, a little boxed essay about creatine. Read about it in the digital textbook as well. And Tuesday when you come back, I have a worksheet all ready for you to help you review this because it will be on your test. So I hope you guys enjoy your 3-day weekend. I hope you do pay attention to this video, and I'll see you on Tuesday. Bye.